I will start this presentation um, introducing you to our technology, Spatial OpenST, that is, as you said, a new method of spatial transcriptomics that is flexible, affordable, and eventually scalable for 3D application. And me and Daniel, together with Marie, and other collaborator, we could develop this method at the MDC, Nicolaus Trajewski Lab. So first, a brief introduction. I don't know if I can move. Okay, thank you, Daniel, for moving the slide. So, as you know, um, since a decade or so, we are seeing an explosion of spatial transcriptomic methods. And now we are all aware that retaining the spatial information of cells is important to understand molecular phenotyping and how this affects the tissue function. And also, spatial transcriptomics is important that because it's possible now to analyze difficult tissues such as adipose tissue that were not possible analyzing correctly due to dissociation problem with single cell technology. So we can mainly divide we can divide the spatial transcriptomics into main categories that are the imaging based and sequencing based approach. Regarding the imaging base, this is one of the approach that is that come with the high resolution that is at single molecule level. However, it it's based on a panel of gene, so it means that you need to have a previous knowledge of the tissue before you start your experiments. So it's more suitable for hypothesis testing studies. In the other hand, we have sequencing-based approach that capture the whole transcriptome, so are more suitable for hypothesis generating studies. However, despite several technology are available on market, sometimes with this technology we have low resolution and also both basically came mostly with high cost and are difficult to implement in standard laboratory due to the equipment and expensive equipment you need to have. So that's why we propose a new technology, OpenST, to bring together the high resolution and also the possibility to have an integrated histological staining, all maintaining a really low cost. And we also wanted this technology to be open. Meaning that is achievable, approachable for any lab in the world. So that's why we provide end-to-end workflow that start from the capture of the RNA in a tissue uh, and with 3D virtual block reconstruction of tissue. So, as I said, the cost is very accessible. Indeed, we reach lower than 150 euro per section without sequencing cost. And this is because we start with capture areas that are made by NGS flow cell. We just do a first sequencing which we recorded the spatial position of barcoded oligo. Then we open up the flow cell, we cut in several pieces of different dimension, and we treat in order to allow this oligo to be to capture the polyadenylated mRNA. Then we place a tissue on this capture area. We stain in order to retrieve histological information. We further treat the tissue to release the RNA in situ, prepare a library for NGS sequencing or other sequencing, and in the end, we combine the sequencing results with the first barcode sequencing in order to have a spatial mapping of each transcript with the subcellular resolution. And then we align and segment using the histological information, and eventually we can reconstruct to 3D virtual tissue block. Now, okay, so we said that this platform is open. Indeed, in addition to our paper, we also write a step-by-step STAR protocol in which you can also find supporting videos. We also have a website that we try to update constantly that you can find this in this following this barcode and QR code. And we also have a discussion board in which we encourage researchers to share their usage of OpenST, but also troubleshooting. Then I will just give you some tips to have some tips and best practice for creating high quality of experimental data. We have several steps for check your quality control. Some are before you begin, and they start with the generating a high quality of capture areas. You can check the quality of your capture area looking at the QC from Illumina sequencing that should have high percentage of passing filter and quality control standard. Moreover, you need to have this sequencing per base sequencing content that of course resemble the base content of our barcode. When you choose the tissue, then you want to have also high quality of image and high quality of morphology. So you should be careful when you embed your tissue in OCT to avoid ice freezing artifact. Moreover, you should check your RNA content and quality, evaluating tissues that reach RIN score more than 7. Then during library preparation, you also have few steps for quality check. For example, when you amplify, before you amplify your library, you can define the number of PCR cycling just by qPCR to avoid overamplification. And then you have another step in which you check the shape of your library and the possibility to have artifact that you have to clean before sequencing. Then in the next slide, I just want you to remind that you have the possibility to use histological images. So it's good if you can take a good image of your tissue, because you can use later for validation, for example, to compare with pathological annotation, but also is important because you can use for segment your single cell to also to select some specific region of interest in your tissue and deepen your analysis. Now for the quality control related to the analysis, I will let the floor to Daniel. Thank you, Daniel.
Thanks, Elena. Thanks so much. So, yeah, before going into this quality control and what we can actually do with the data, I just want to say a few words about the computational workflow. So, of course, we made all of these tools open source and widely available to the general public. And as Elena mentioned before, the main computational workflow consists of three major modules. First one that takes care of pre-processing and data analysis on the imaging side. Module B that takes care of transforming your transcriptomic sequencing output into digital objects where you have gene expression at each of these 0.6 micron spots. And then module C that combines the two modalities into a single object. So at the end, you get a digital gene expression per each of these segmented cells, and of course, with the respective spatial coordinates and any other metadata that might be related, for example, to cell morphology. And already as mentioned, if you have an experimental design where there is consecutive sections or sections at least from the same tissue, we also provide the tools. This is, for example, the STEAM algorithm developed in collaboration with the Stefan Prebisch lab, where you can use this data in order two-dimensional data can be reconstructed into a 3D virtual tissue block, which is a digital object that lives on your computer, where you have a combination of both the histology of the tissue provided by the imaging and also the whole molecular landscape in terms of messenger RNA that is expressed in each location with subcellular precision. So after, let's say, following these computational steps, and before moving on to the exciting part of data analysis, we of course have to make sure that our data is good enough. And there is three major criteria that we follow in order to decide whether a sample is good or not, and we should proceed with the data analysis. First, we take a look at the quality metrics of the sequencing side things. And here are some numbers that we would consider good enough. So a sequencing output that provides around 5 to 10% ribosomal RNA is usually expected in OpenST. And then among the reads that are excluding ribosoma, you get more than 70 or even 80% of uniquely mapping to the genome. And among those, you have an almost half and half distribution between coding and 3' UTR transcripts. So specifically to say that we do have a 3' bias like other technologies, for example, 10x single cell sequencing. Also, as mentioned by Elena, we also make sure that the imaging quality is good enough to perform single cell segmentation, so we can take advantage of this multi-model integration and creation of single cell profiles by using the images as a proxy for segmentation. In case the images are not good enough, we also provide additional computational tools based on deep learning algorithms, generative models, to enhance the quality of these images that down the line also improves the quality of visual assessment, but also cell segmentation algorithms. And more critically, one needs to make sure that the alignment between these two modalities is as precise as possible. So we do provide these automated or semi-automated tools to perform the alignment, also to validate the quality of the alignment visually and quantitatively. And as you can see there, even small deviations from perfect alignment, even like only 5 microns, already breaks the subcellular enrichment of very specific nuclear or cytoplasmic markers. So having these three is essential if you want to do the full set of possible analysis that can be done with OpenSC. Namely, you can, of course, analyze gene expression in spatial coordinates, even without images, and also having this sort of distribution of features, like coding features in the OpenSC data allows you to not only do gene expression, but potentially even some isoform calling, in particular from 3' UTRs. When you have different degrees of image quality, you can range from cell counting up to cell morphology analysis. And when you do have a very good alignment between spatial transcriptomic data and your imaging data, you can go from both cell calling, but also really perform subcellular enrichments of individual transcripts. And we wanted to put our technology to test. We didn't just generate or create OpenST in a vacuum. We had many questions in the lab, systems biology questions, where we actually need to have a systematic profiling of single cells in a very deep way, so we can recover the whole molecular landscape of these single cells, also preserving the architecture of the tissue. And one of these big biological questions or problems that we wanted to address was going back to clinical samples from Charité. We wanted to really understand tumor heterogeneity, in this case, from head and neck squamous cell carcinoma, and more particular to understand the establishment of cell type heterogeneity, tumor cell type heterogeneity in the primary tissue, and how this is maintained or not in the metastatic tissue, in this case, a lymph node. So we did indeed sample these patient samples from the Charité. It's a large university hospital here in Berlin, one of the largest in Europe. And we actually explored 3 questions that can really take advantage of OpenST as a whole. First of all, about cell type heterogeneity, and this is something that you can also do actually with single cell technologies, not only discover major known cell types, but really go down into specific molecular subtypes of cells that can be potentially novel and driven by, for example, new gene programs or gene modules. But we do have spatial resolution, so we can now start to answer questions that single cell technologies cannot. And those are questions related to, for example, microenvironment modeling, how specific cells communicate locally, and also the very essential questions of how are or what are the molecular programs that are established at the very critical boundary between tumor and healthy tissue. So we indeed took these sections from these 2 tissue blocks, one from the primary and the other from the metastatic lymph node from the same patient. And when we performed OpenST protocol, we were very happy to see that we got super high quality data. So long story short, we got most of cells with around 2,000 to even more than 4,000 unique molecules and genes captured per segmented cell. And not only very deeply sequenced cells, but also lots of them per each of these tissues. So we also have very minimal losses of cells within the tissue. We preserve most of them, with 90% of them with high quality. This data, so I said is between 2,000 to 4,000 unique molecules or genes per cell, might sound slightly lower than your typical single cell experiment. That's true, not because of a limitation of the OpenST protocol by itself, but because of a limitation in sequencing depth. So here we sequenced every cell to say 10,000 reads per cell. That's actually 5 times lower than the sequencing depths that you use for single cell data. Problem is that here we have lots more cells that you get on your average single channel, let's say 10x Chromium run. And we know that we could push this data even further, because even when you compare to other technologies, OpenST is amongst the most efficient in terms of cost per amount of information that you get, in this case measured by the amount of reads that you need to invest in order to discover new unique molecules or genes. So both for these datasets, but also for others that we used in our paper, like mouse tissues for benchmarking, we know that we could sequence even deeper, and we are still far from saturation, so we still have the ability of really discovering new molecules from the sequencing libraries. Anyway, we took the data that we have, and we anyway managed to perform cell typing as expected. So we just used the typical algorithms that one could also use for single cell analysis. It's the profiles we have both in terms of the single cell resolution, but also the depth were comparable enough. And we recapitulated the major cell types that one expects to see in these primary and metastatic tissues. Remarkably, when we project these data to spatial coordinates, we can see not only the major big compartments forming, for example, or formed by tumor cells, but also we can see cell states that are very sparsely distributed across the tissue. In the case of the primary, very sparsely distributed cancer-associated macrophages that would be very difficult to capture with single nuclei data or single cell data. And also very remarkably, in the case of the metastatic tissue, we could recapitulate lots of the molecular and cell type diversity of the very complex and challenging also to work with lymphoid part of the metastatic lymph node. We validated this sort of cell typing with an orthogonal modality. In this case, it was an imaging-based protocol. Xenium from 10x is a probe-based protocol. In this case, we use the version of Xenium with 350 genes. And when we compare the output of cell typing, at least on a basic level of cell typing between Xenium and OpenST, we could see that the two modalities matched very well. So that was a really nice to have orthogonal validation. I'm showing here primary, but also same applies to metastatic lymph node. But even when we subset OpenST data to the shared subset of genes, that would be actually 1% of the data that we have from OpenST experiment. We still see very good agreement between the two modalities, telling you that also, as we can see from a bulk level of gene expression, and also when projected back to spatial coordinates, the two modalities agree very well across the whole range, the dynamic range of gene expression. Anyway, the main power of OpenST compared to, for example, to Xenium, even when the cell types, major cell types are the same, OpenST has 19,700 more genes, plus additional long non-coding RNA that we can capture. It's basically unbiased versus a probe-based method. So one of the main powers of OpenST, as I said before, is that you can discover potentially novel molecular states or subtypes of cell types. And that's actually what we did for the tumor compartment. So we isolated those tumor cells across both the primary and metastatic tissue, and we performed joint subclustering, embedding and subclustering of these cells across the two tissues. We indeed discovered 10 molecular subtypes of tumor cells that in the case of the primary tissue were strongly organized in spatial domains that also followed the composition of the stromal compartments and were more disorganized in the case of the metastatic, not following a particular structure except for some few of these molecular subtypes. But we did not just get some discrete states of tumor cells, but more importantly, since we do have this gene information, we can annotate what are the actual molecular programs, molecular pathways that are explaining that discrete set of tumor subtypes. And we saw an amazing diversity of different gene programs ranging from very proliferative or different terminal states like super invasive and aggressive pre-EMT or less aggressive keratinizing cell states, also having some cholesterol biosynthesis or more inflammatory, inducing or responsive tumor cells. So this shows you that you don't only get the name of the cell type, but you can also characterize very deeply and potentially find even new gene modules that explain the specific phenotypes and molecular pathways that certain specific single cells have in tissue context. We also wanted to understand a bit more about the biology of these molecular subtypes of the tumor cells. So we, of course, started to look into what's the interplay between these substates of tumor cells that were seemingly so spatially organized and the stroma. And for that, we took a look at the communication of these cells together with the stromal compartment. For that, we use some spatially restricted ligand receptor analysis, which basically takes a look at RNA expression of ligands and receptors from a database. And that can be used as a way of predicting what is the activity of cell-cell communication in, from this sort of single-cell spatially resolved data. We have lots of these ligand receptor pairs. So one way of summarizing and looking at the data is to, for example, perform spatial non-negative matrix factorization. So instead of looking at thousands of individual cell-cell communication programs, you can look at, let's say, factors or motifs, spatial motifs of cell-cell communication. So you get non-negative scores for each of these cell-cell communication events, and they can form specific patterns or archetypes. And when we look at these in space in comparison with the organization of these tumor subtypes in space, we do see that there is a very good agreement of some of these communication hubs or archetypes co-localizing with very particular, very specific tumor subtypes. For example, pre-EMT phenotype seems to be very well correlated to the presence of a matrix stiffness cell-cell communication, mostly dominated by laminin and integrin mediated signaling, and also the inflammatory inducing tumor cells were also in agreement with inflammatory response from the immune system. And this cell-cell communication was indeed happening between the tumor cells and stroke. Not only cell-cell communication, but also we can take a look at other interesting spatial components, like, as I mentioned before, what is the interplay between healthy and tumor side? This is something that we could only do in the metastatic lymph node, because unfortunately we didn't have a healthy counterpart of tissue in the primary section. It was all full of tumor. But we do have this opportunity in the lymph node where we have the basically normal human lymph node structure living together with the invading tumor. And we computationally now can define what is the exact boundary between these two pieces of tissue with cellular or even subcellular precision. And we can now ask the computer what are genes or gene programs that are enriched at either of the two sides of this boundary, again, with single cell resolution. And just to show you an example, when we took a look at these programs, both from the lymph node side and the tumor side, we found on the lymph node side activation or recruitment of T cells mediated by activated macrophages. And on the tumor side, we saw a very interesting signature for cholesterol biosynthesis that was indeed very active on the boundary between the tumor and the lymph node. And we can validate this not only in 2 dimensions, but we do have these virtual tissue blocks that we can explore interactively. And we use this ParView application that is usually used in engineering or physics. And we basically repurposed it to a super interesting, super nice browser of 3D data. So this that you see now is the combined in the same basically 3D objects, the expression of this CXCL mediated signaling for T cell recruitment and this cholesterol biosynthesis. So you can there see how really the cholesterol biosynthesis pathway is really all surrounding the tumor in 3 dimensions. And this T cell recruitment mediated by macrophages, this signature is also living next to the cholesterol biosynthesis pathway in a way that it's also 3-dimensional. So this is all super nice, and we can show, at least to some extent, these are some basic analysis on what you can do with RNA data resolved in spatial coordinates. But there are some things that we are leaving out. So this is just the classic OpenST, but there is some information that would be nice to have. And now we are working on extensions of the protocol to have this additional information. One of them is to include, of course, deep single cell and single RNA sequencing data. As I said before, we are a bit limited sometimes by the sequencing power. So we don't want to maybe sequence too deeply, but we can still rely on already existing single cell data in order to improve the annotation of our cell types or even project other let's say pseudotime or velocity scores that you can compute from these sort of dissociated cells. But also we would like to take a look at other non-human, potentially non-human RNA, like to study the interactions of host and microbiome, in this case of bacteria. And more importantly, to look at other layers of gene regulation. So not only stay at the level of RNA, but also take a look at, for example, proteins. And in the last 2 minutes, I will just show very quickly examples of this. So for single cell data, why we actually integrated with OpenST, this is a very interesting preprint that I recommend that you take a look at it from 3 colleagues in the lab, Camille, Ilan, and Tancredi. So the main question that they had was whether it's possible and how to, for example, trace tumor cells from the very cell of origin that looks healthy until the very late stage. And they leveraged a mouse model with 3 mutations in oncogene, some loss in function, some gain of function. Long story short, these mutations make that specific cells at a very specific stage of the mouse development, can create asynchronous events of tumorigenesis in the mammary gland of these mice. And when you profile these at different time points, even within the same sections, you see asynchronous growth of different tumor and that are isolated spatially, even within the same time point and same section. So when you generate single nuclei and OpenSC data, from the single nuclei data, you can do very precise profiling of what are the pseudo-temporal trajectories of tumor cells from the healthy to the very late stage and annotate the molecular phenotype super well. But having the spatial data allows you to project the single nuclei data into space. And what this gives you, in short, is a space-time resolved map of tumorigenesis, again, from the very early events to very late events, which allows you to get a data set that looks like this. So you get more than 100 snapshots of tumorigenesis, spatio-temporally resolved, from very early events to very late events. So you get the temporal progression of not only the tumor cells, but also the tumor cells together with the microenvironment and how these get remodeled and what signals could be intercepted as early as possible, for example, to avoid tumor progression. So if you want to know more about this data, then I really encourage you to go to the preprint. And again, in the last minute, just to say a few words about spatial host microbiome analysis. So one of the modifications that we can apply to the OpenST protocol is to replace this poly-DT oligo that captures polyadenylated messenger eukaryote RNA by other sequences. For example, in order to capture ribosomal RNA from bacteria that would allow you to characterize what species you have in a piece of tissue. So if you put the sequence of that 16S constant region of the bacterial 16S RNA, then you can actually capture it with the same subcellular resolution, and then you can get these maps of not only gene expression, spatially resolved gene expression, but also what species of bacteria you have in specific subcellular locations on your tissue. And in this case, we use a molecular genes design, which is, of course, known to have lots of bacterial diversity as a nice control to start to explore this new type of technology. And in the sense of proteins, now we have some modifications to the OpenST protocols and extensions, so we can not only perform H&E staining as we do for our typical cell segmentation, but additionally now you can add, for example, DAPI staining, membrane staining, but also interesting proteins that you might be studying. Like in this case, our colleagues Miriam and Anastasia, they are interested in human brain organoids, and there are some delays between the expression of protein and expression of mRNA, and this, in the case of this organoid that has a very specific, interesting layering of these phenotypes. So they really wanted to study this agreement between protein and mRNA, and the cool thing about this protocol is that you do this staining and mRNA capture on the same section. So you can really study the same cells in a very multimodal way, having this imaging data, H&E, but also now protein information. And you can start also to relate protein levels, not only to the messenger coding for that protein, but potentially to other messengers. This right now is limited by the typical IF protocol that only allows you to do a bunch of proteins at a time. But as the immunofluorescence methods improve, and then we can also enable multiplexed IF, then we will also improve the ability of capturing multiple proteins together with multiple messenger RNAs. And just as an outlook, last slide, this is how we are currently leveraging OpenST. So I've shown you how we can explore known mechanisms in space, how we can discover potentially new correlations or structures within the data, for example, to define very new or previously unknown cell-cell communication hubs and communication motifs in space. But we are also now leveraging this data to, in combination with, for example, drug activity databases or drug target predictions, to understand about the effect of specific drugs, not only in a bulk sense, in a tissue or in a patient, in a systemic way, but really pinpointing what would be specific regions of the tissue that would be reacting the most to specific drugs. And as you can see from that example, if we take an already existing package like this drug to cell from the Teichmann lab, now you can start to really make predictions of where in the tissue a drug would be more active. And we did this on our primary tissue and made a survey of different drugs that could react in specific parts of the tissue. And for example, here you see that this drug D is the one that would have the most strongest effect all across all tumor cells, whereas drug B would only react to very specific subset of tumor cells, at least based on what we can predict from gene expression. And of course, all of this wouldn't be possible without all of the effort of a very large group of amazing people from the Rajewski lab and also the groups of Pino Macino and Elisabetta Ferretti, both at MDC and Sapienza, many collaborators that both provide samples, but also great intellectual exchange. And also thanks to the organizers of this seminar for giving us the opportunity to speak today to you. Now we are really looking forward to the discussion. Thank you.